then followed by Rajesh Pai's lecture on the fuel chemistry. He talks on the metal organic framework and how they are useful. So in today we have covered many eminent scientists, and now today we are at the end of this lecture series, and we are very much fortunate to have the Nobel Laureate in the last as a talk of him. Now I request Professor J.V. Acme sir to discuss and give the small introduction of Professor Jeamn-marie Lehn.
Good afternoon and good morning, Professor Lehn, because you have.
Yeah, good afternoon for you.
Yeah. You are 10:30, I guess, and we are 3 o'clock here. We all feel sitting here, I'm addressing the audience, we feel delighted and thrilled to have with us Professor Jeamn-marie Lehn, who won the 1987 Nobel Prize in Chemistry for his work on synthesis of the molecules called cryptands. I'll tell you what they are, cryptands. But even before getting the Nobel Prize, Professor Lehn, whose career I have always marked with great enthusiasm, he was known as someone who started the field of supramolecular chemistry. Supramolecule means large molecule. This is a typical thing, wording. In fact, he introduced this word supramolecular to the rest of the world. We in chemistry and physics were not using this word earlier. So, supramolecular chemistry. And this is chemistry of something like host-guest chemistry, chemists understand, using molecular recognition. A molecule can recognize the other molecule, so they get attached. It's a bit complex, but we'll hear from him. In 1980, Professor Lehn was elected to become a teacher at the prestigious Collège de France. Means you are a member of Collège de France. At any given time, from a given subject, chemistry, physics, or whatever, only 10 to 12 French scientists of physics or of chemistry are members of Collège de France. It's a great honor which he got in very early part of his career. Now, a cryptand molecule I mentioned, it's a ligand that can bind structures in a crypt. Crypt means gadda. It's a pit, it's a cage. So the molecule can get trapped in that. These molecules are three-dimensional analogues of crown ethers. Let me not go into details, but they are very selective and they can even trap cations. Sometimes it's very useful to do that, like potassium ion. I used to read about supramolecular chemistry and the work of Professor Jeamn-marie Lehn. And also I used to read in early 80s, the lock and key principle, again, molecular recognition, which is a principle of the action of enzymes, organic catalysts. So once, after he got his Nobel Prize, I got very excited. Once I was in Bordeaux, spending some time in 90s. So I told my host, late Professor Olivier Kahn, and Professor Lehn knows him, I want to meet Professor Lehn, I told him. He says, it's simple. I go every Monday from Bordeaux to France. I am a member of Collège de France. I go there, deliver my lecture and come back in the evening. You come with me. I'll introduce you to Professor Lehn, and then you can ask him any set of questions. That floored me because I had no questions to ask. I told him, we typically Indians, we like to, like touch a great man, feel the presence of a great man. It is that. I got a chance to do that later in 2014, when in Valencia in Spain, we were all together and I could talk to him. Then I got excited, and I was just mentioning before 3 o'clock to him, some of you were hearing. I was in charge of the Atomic Energy Central School, and we had a video recording conference room. So I went to
chemistry is one of the very important sciences which shows which steps the universe and matter in our universe is going through to become complex. Now, in order to put it in a broad perspective, we have to start very, very early. 0.7 billion years ago. What happened then? What happened is the following. There was the Big Bang and our universe started. Let me just insist on one thing, which is quite interesting. Rather recently, by measuring the background, that the theory of inflation was indeed correct. That means that at the beginning of our universe, there was a very, very rapid inflation. It was so rapid that it was 10 to the minus 36 seconds and 78 powers of 10, 10 to the 78. That's an enormous inflation which shaped our universe at the beginning. Now, after that, of course, the universe was much too hot. There was no chemistry at that time. And particles formed, the particles went together to atoms, and this was the age of physics. There was no chemistry at that time. The conditions were not correct for having chemistry forming, because when atoms formed, then after that, molecules could form. And at that stage, chemistry could. That is the age of chemistry.
Yes.
Now, once molecules were generated by combination of atoms, then they became more and more complex. They changed, they came together, they determined bigger and bigger molecules. They also generated layers, which are sort of primitive cells, so that after that, what happened is that life appeared on our planet, Earth. We are sitting on this planet, and this phenomenon we call life, we know it around us. We are part of it. But there is certainly life in other regions of the universe. That is the age of biology. And most importantly, these living entities developed more and more faculties, possibilities to not be here. Thought is what characterizes mankind and maybe other entities around the huge universe. I represent this thinking by this famous statue of Rodin, the thinker sitting there. And this, of course, is then the steps towards more and more complex states of matter. What we know on our planet, the most complex is the human being and the thinking, the brain. Let me also insist on one thing. As you can see, this is the end of the slide here. That's the end of the slide. But it's not the end of the evolution of the universe. The universe will continue to evolve. It will become different. It will change. And nobody can stop that. So when people come around and tell you, we want to stop it, we should not continue, this is not possible. Thinking is here, life is here, and more complex states can appear. So we cannot stop evolution, not at all. And the universe does it without asking us whether we want it or not. Now, the universe, in fact, is for the moment still something we don't understand entirely. This slide is not a bad slide. This slide is just, I wanted it this way. Why? It shows the following, that the universe is made up of 68% of dark energy, as cosmologists tell us, and 27% of dark matter. That means it's 95% darkness. All right, that is not very encouraging. But there is 5% of visible matter. We are part of this matter, and this is what I call the matter that matters. That's our matter. That's the matter we are part of. And that is the important matter for us. Now, what is then the evolution? We start with divided matter, particles, atoms, then it condenses, it becomes organized, making primitive cells and then an organism, then living, thinking, and maybe even something more complex beyond our thinking matter. So as time goes, matter has increased in its complexity, and this complexity is sort of related to more and more information in matter. We will come back to that. There will be this will be an important point. Maybe there are more complex forms of matter than we know. Maybe in other regions of our enormous universe, there are already more evolved forms of this complex matter, but this we don't know. So this is the step. These are the steps towards complex matter. And one may say that it looks like the matter has evolved under the pressure of having some regions of the universe becoming more and more informed, more and more complex. Then the big question we have to ask, is the following. How does matter become complex? What are the ways in which matter has gone from elementary particles up to a thinking organism? And maybe there are even higher forms of complex matter. Just to illustrate that, mankind has developed what we call science. And if you look at three main domains of science, physics is this part of science which deals with the laws of the universe. These are the laws on which everything else depends. These are the basic laws of our universe. We depend on it, everything depends on these laws. Biology studies the rules of life. These are not laws. The laws are given by physical laws. Biology is the way it functions, the way living organisms function. So what is chemistry doing? Chemistry has a very important mission, which is to build the bridge between the general laws of the universe and the specific expression of these laws in given complex entities like living organisms on the planet Earth. Don't forget, the planet Earth is just a little piece of dust within an enormous universe. But the important thing is that it has led to the development of this complex matter, which results from the laws of the universe. Now, what is the answer to this question? One can define the answer to this big question, the most important question to ask. There's no more important one. It has become complex by self-organization. Now, this is not really an answer. It just says that matter has become complex by itself. So the laws of our universe are such that life, thinking, has developed spontaneously due to the structure of our universe. One can even go a bit further and say that it is a cosmic imperative. In our universe, the way it is, the fact that organization occurs is for me not an accident. It's not hazard. It is because it is built in. It's an imperative. So I am quite convinced that there is life and thinking in other planets in our universe. On other planets, there are other organisms which think, which live, which think. And of course, it would be very interesting to meet them someday, not now. Now, chemical evolution of matter was at the beginning not at all a biological evolution. It was not a Darwinian evolution. It was an evolution driven by self-organization of non-living molecular matter. Just electromagnetic forces, the physical forces acted on the bricks of our universe, the atoms, and make these combinations of atoms more and more diverse, more and more complex, and this generated then these molecules which contain an information, as we will come back on. So that by Darwinian evolution is a subgroup of evolution. Darwinian evolution is evolution of living matter. So it is self-organization of living molecular matter. In fact, chemical evolution is much wider. Darwinian evolution is the evolution of living matter, but chemical evolution is much broader. It encompasses non-living and living matter. In the old times, if we go back to the philosophers of the country in Greece, in Europe, 2,500 years ago, like in other regions of the world also, people were just thinking about what is this matter, what is around us. So for instance, there was a philosopher at that time, 2,500 years ago, called Empedocles. He had a simple idea. He said there are 4 elements in matter: fire, air, water, and earth. And the combination of these elements gave properties: dry, hot, humid, and cold. So that was a start. But at the same time, there was somebody who had a much deeper insight into what matter could be. This is Democritus. Democritus said 2,500 years ago that matter is made of particles which cannot be made smaller. And this is, in Greek language, atomos, atoms. That means entities which cannot be cut into smaller things, which are the building blocks of matter. This was 2,500 years ago. So Democritus had already the clear representation of matter, which is that matter is particulate. Matter is atoms, bricks, with a lot of void between them. It is said that Democritus had this idea from some experimental, or let's say observational data, which are very simple. If you go around your city, Aurangabad, and want to convince somebody that matter is made of particles, matter is particulate, you can do the following. You take a piece of salt, you put it into water, and what happens? It dissolves. In other words, the piece of salt or sugar, when you put it into water, dissolves and disappears in the water. This can only be explained if you admit that both water and the piece of salt or sugar is made of particles which can mix. If they were continuous, it would not be possible to have one disappearing in the other one. So this is said, this one thinks that this was Democritus' reason to think that matter is particulate. And it was, in fact, a fantastic step in trying to understand what matter is made of. Now, about many years later, in 1869, there was a big change. People had progressively accumulated the notion that matter was made of elements. We come back to that, of bricks. But these bricks were not very organized. It was a bit chaotic. Until in the middle of the 19th century, some people came up with the idea that one could perhaps make that more rational. And the most successful was Jeamn-marie Lehn, who in 1869 published a paper which you see here. That's the original paper, which is the report by Mendeleev in German language at that time of the fact that he was able to organize the elements, the bricks, which at that time were known in a way which you see here. There are columns and there are rows. Right now it's turned around. As you will see, the modern table of the periodic table, but the idea is exactly the same. It's just that what is here in columns is now in rows. but this is only a way of representing. So here you see the original first publication of the periodic table of the elements. It organizes the elements according to their properties and their atomic weights, Eigenschaften. Eigenschaften is properties, Atomgewichten is molecular weight, is atomic weights. And when you do that, you end up with this. In fact, Mendeleev went a step further. In some of these, when he put this table together, there were places where there was a void. And he put here a question mark, as you can see, and he said, someday we will find these elements, and they were found. So that was a fantastic step in understanding what makes up visible matter in our universe, the 5% of matter. Nowadays, these relationships are described in a periodic table of the elements, which in the modern representation is this. The bricks of visible matter are shown here. Now, when you go to chemists in the room, they all know that. That is classical for them. But do they really realize what this means? When you look at that and think what it represents, it is absolutely fantastic. What you have before your eyes is the series of bricks which makes up the table, which shows the bricks which make up our universe, the visible matter in our universe. I can even add, there are no others. These are the natural elements. This is our universe. And the visible matter in our universe is made of elements from hydrogen here to uranium, which is the here. These are the natural ones. The other ones, the here, are transuranians. That means these are made in accelerators. They are artificial elements. But let me really insist on that. This is absolutely incredible that mankind has been able to put these elements, these bricks of visible matter, into a table which is full. There is no other one. I think this sounds terrible because it sounds like it's a belief that it is some kind of a dogma. It is not a dogma. It is just reality. And why is there nothing else? Because if you count 1 hydrogen, 2 helium, 3 lithium, 4 beryllium, and so on, these are elements where you start with 1 proton in the nucleus, 1 electron, then 2 protons, 2 electrons, 3. Of course, there are isotopes, but let's forget about isotopes. They don't count really. And between 1 and 2, 2 and 3, 3 and 4, 4 and 5, and so on, there is nothing you can put between. So this table, as you look at it, I really insist, I'm sorry to repeat myself, but I think it's so incredible. This, what you see before your eyes, is what makes visible matter in our universe. And if you look at a very far distant galaxy, they will be the same. And what the combinations of hydrogen and oxygen, for instance, making water is the same everywhere, billions of miles away. So I think this is an incredible step in knowledge which has been brought about by these 2-legged animals on a planet called Earth. And this is the playground of chemistry. Chemistry plays with these elements to construct more and more complex entities, takes these atoms and makes out of them, according to the laws of physics, what you are allowed to do, but by physics, physical laws, and makes out of them more and more complex molecules. Now, so when using atoms, you can make molecules. In other words, from bricks, you can make houses. And this is the development then of molecular chemistry, which results from combining atoms by strong bonds which link them together. Chemists call these covalent bonds, which allow you to cement these atoms together and to build more and more complex molecules. Let's show you 2 milestones in molecular chemistry, just to give you an idea. And it is now accepted that it started with the production, the fabrication in the laboratory of a molecule called urea, which you see here. This is contained in urine. It's a rather simple molecule. Why do I show you that? First of all, because it was making urea from another molecule, ammonium cyanate. Why do I mention that? Because that shows also the power of knowledge to change completely the way in which we look at the world. At the time of Wöhler, people were thinking that you could not produce in the laboratory a molecule contained in a living organism without a magic force called the vital force. When Wöhler made urea, which is contained in urine, from ammonium cyanate, he made the molecule contained in living organisms without the help of this magic force. So that magic force does not exist. It just means that the world of molecules, be they molecules of non-living matter or molecules of living matter, that's the same. There's no difference between what is life and what is not life. Now, about, as you can see here, 150 years later, there was a production in the laboratory of a very complex molecule, vitamin B12, which you see here. That's a vitamin you have in your blood. And this vitamin is much, much more complex than the simple molecule of urea. So in these years, the evolution of these years, the chemistry has become very powerful, able to produce in the laboratory this very complicated molecule called vitamin B12. This was done by two groups working together, collaborating. One was directed by Robert Burns Woodward at Harvard University in the USA, and the other one by Albert Eschenmoser at the ETH, the Polytechnical School in Zurich, in Switzerland. And they made this with the help, of course, of many collaborators. There were probably, I don't know exactly how many, but probably 100 collaborators. I worked with Woodward as a postdoctoral fellow, and this is my part here, which I made of this molecule. I don't show that just to show that what I did, but I wanted to show it, that it is a sort of an illustration of science, the fact that if you contribute something to science, you bring your stone to the fantastic building of knowledge. And so you contribute your stone, and your stone is yours. Nobody can take it away from you. So molecular chemistry continued to evolve. It was more new reactions were discovered, new compounds were made, new materials, and so on. So this is still a very, very varied, very active science. It's still a lot of things to be discovered. And one has now many more possibilities to make drugs, to make new materials, and so on. But then you can ask a question. Should one not look at something else? Is there some other type of chemistry, which is part of chemistry, of course, but a part of chemistry which one should have a look at? I illustrate it by this biological example. Here you have a cancer cell, the blue sphere, and two killer cells in purple. The killer cells have a mission. In your organism, as you are sitting there, they are the policemen or policewomen, whatever you want. They run around, they try to find out which cells have become bad, transformed, have become cancerous cells. And when they find them, you have to destroy those cells. So how do the killer cells, to these purple bodies, know that the other one is a cancer cell? They are defined by a membrane, which is made of molecules. And in this membrane, there are other molecules which sort of tell the killer cells that the other one is a cancer cell. The same thing can be said of this. This is a white blood cell. With the blue dots is a HIV virus. And when the HIV virus gets in collision with the white blood cell, it now knows, if I may say so, it knows, that it's just a way, of course, of speaking, it is now able to infect the white blood cell. The same processes are all over in your body. For instance, antibodies and antigens, vaccines, and all that. These are molecules which recognize other molecules. So there is something very basic here. These large entities, much, much bigger, of course, than molecules, are made of molecules. They are able to perform a recognition process which tells one entity what the other one is. And this is, of course, fundamental for living organisms. So molecular chemistry started in 1828, and we proposed in 1978, when I introduced that name in 1978 in 2 papers, which is supramolecular chemistry, which is a chemistry beyond the molecule. Of course, supramolecular chemistry does not exist without molecular chemistry, and supramolecular chemistry is a chemistry which is a sort of a trying to understand what populations of molecules are doing, mastering the interactions between molecules, and then trying to understand how this what are the features which result. Three main properties were studied over the years. The most fundamental one is molecular recognition. How do molecules recognize one another? Then, how do they react when they get together, to transform each other? And also, how can they go through a barrier, a membrane, like a cell membrane? These are transport processes. But let's have just a look at one, which is the most basic one, molecular recognition. What is molecular recognition? Without molecular recognition, we do not exist. We cannot function without molecular recognition. So what is it? Molecular recognition first requires interactions. Things have to interact, to bind together, otherwise there would be no effect. They would just ignore each other. So these interactions are very important. But there's another property which is important, which is information. When you talk of molecular recognition, you must admit that there's information present, because that's the way to recognize somebody, have information about a person. So there's a molecular recognition, a recognition between molecules. And one can represent that by saying that there's a sort of a complementarity in geometry and interactions. They have to fit together. The two objects, or more than two, have to fit together in geometry and in their interactions. That means, for instance, plus attracts minus, and plus repels plus. Now, this idea of molecular recognition was also already presented in very famous paper of 1894 by Emil Fischer. Emil Fischer was a fantastic organic chemist. He was probably one of the greatest ones. I would say he's certainly one of the greatest ones, maybe the greatest ever. And he said in a paper in 1894, studying the action of enzymes on their substrates, that in order to act on each other, molecules have to fit together like Schloss und Schlüssel, like a lock and a key. This is the basic concept. It's a very simple one. Of course, it's too simple, so to say, to describe the complexities of living organisms, but basically it is correct. Molecules fit together, and this allows them to act on each other. You may note that Emil Fischer got his doctorate in our university in 1874. And of course, we are very proud of that. These were other ages. We are now in Europe. At that time, we were German. Now we are French, but of course, we are all in Europe now. Now, the prime example of what molecular recognition has as role, the role of molecular recognition, is the famous double helix of DNA. This is the molecule which contains the genome, the genetic information of all living organisms. The storage of this information resides in a very simple process, in a very simple way to store information. There are 4 groups. These 4 groups you see here, for those who are biologists or chemists in the room, of course, they know all that, but I'm speaking here for the others. There are 4 chemical groups: adenine, guanine, thymine, and cytosine. These are like 4 letters. The names are given by chemists just to represent them. And these 4 letters are appended to a long molecular chain contained, formed of a sugar and a phosphate. Let's forget about that. It's like a string on which these letters are hanging, are connected. And it is the sequence of these letters which makes up the genome of living organisms. The sequences are very specific, and this makes the difference between an elephant and a tomato, or an elephant and a human being. So this is the storage of molecular information in the genetic program. Now, storing is not enough. You have to be able to process the information, and this is done by pairing up these letters. You see these letters, these 4 letters, they can pair up to make 2 points of interaction contained in this green rectangle, or 3 points of interactions here. So adenine interacts with thymine or uracil, simple derivative, by 2 points of interactions, and guanine with cytosine by 3. So it looks like extremely simple, but probably that's the reason why there are only 4 letters and 2 ways of pairing. 4 letters is enough to store the information, and so as to read it, to process it by forming 2 pairs. 1 pair with 2 points of interaction and the other one with 3. In other words, whereas in a computer, for instance, the storage is 0101, that means no current, no current. Here it is 232323. And this is enough. It's very simple, but probably evolution in some ways along the ways of development of evolution, there was so maybe other trials, maybe they have at some stage been more letters, maybe other ways of pairing up, but this was probably chaotic, was not feasible, was too complicated, so that what survived is the simplest form. 4 letters, 2 ways of pairing them up, and that's enough to store information on the molecular level and to read and process it on the supramolecular level, because the molecular letters, they make supramolecular interaction with 2 or 3 interactions. Now, chemistry, therefore, how can we define it? What is classical is to say that chemistry is the science of the structure and transformation of matter, non-living or living. That means how it is built and how it can be converted into, how the molecular bricks can be converted into one another. But that's not enough. From what I just told you, at the supramolecular level, there is another notion which is very important, which is absolutely basic, which is that chemistry is also an information science. Molecular matter contains information. So chemistry is not only the science of the structure and transformation of matter, but also the science of informed matter. Molecular storage, it's the in DNA, to take that example, it is the storage, the sequence of these letters, and the processing is the way in which they interact, these letters interact with one another. So it's a supramolecular processing on the basis of a molecular storage. Then, of course, many people started to try to understand how molecular recognition occurs, how to understand this molecular recognition. And here, if we use the terminology of Emil Fischer, we have here a lock here, this molecular receptor, we can receive another molecule. We have here the keys, 3 different keys in this example. And as you can see, of course, obvious, the red key is the one which fits into the lock. So this lock and key process was studied by many, many groups around the world. In many countries, of course, I know many in India also. I know many people in India in laboratories who have been studying molecular recognition in all countries. Now, let's give you just a few examples of applications. Of course, as you know, without basic research, there's no application. So we have first to acquire the knowledge in order to be able to apply that knowledge. So the first and most basic application is drug discovery. What is a drug? If you want to make a deficient drug for a given disease, you have to make a key for a biological lock. In other words, you want to make a molecular key which will selectively bind to another molecule in an organism, which is the lock. And this is the basis of pharmaceutical chemistry or pharmacy products, which we all depend on. Then we also developed over the years, I showed you mostly our results which we have obtained. And there are many others, of course, by many groups. So we also made this kind of unit here, where there's a molecule here in purple, which has a europium ion and metal ion inside, and this emits red light. And on this basis, method of diagnostics was developed here, which is here you see the apparatus. It's an immunodiagnostic system, which is used in many hospitals. Then, what about transport? Transport processes, which I mentioned, are processes by which a given molecule can go through a barrier, a membrane, either an artificial membrane or a cell membrane. And this gene transfer is the process is to take a piece of DNA, a gene, to make some kind of a molecule which may help it to cross the membrane of a cell, and then to go to the nucleus to pick up the RNA, and then to make the product, ultimately will be a protein. Now, this kind of gene transfer is very important for gene therapy, for correcting diseases where genes are deficient, and also in biotechnology. Now, you may note that what I'm just telling you is to make genetically modified organisms. I know many people protest against those. I am convinced that they are very important for us, mankind. They will be made and they will come around. Nobody can stop it. We will need this. We need genetically modified organisms because we need to produce, for instance, food in conditions which different climates, hot climates, cold climates, and so on. And so genetically modified organisms are here to stay, and they will stay. Let me also mention the question of making materials of supramolecular type. That means materials where the entities inside are linked together by the supramolecular forces and follow the rules of these non-covalent, as I call them, forces. Let me give you an example, which is of interest. In 1990, we have introduced in a paper the notion of supramolecular polymers. That means, let's say, in another way to speak, of a supramolecular plastics, let's say, supramolecular plastic materials. And this was done at that time in 1990. We published the first paper in 1990. But then, many years later, which means in 2013, I received information about the first application of supramolecular polymers of biocompatible type for cardiovascular implants. This was quite a surprise for me, because I didn't know this type of work was going on. There was a group in the Netherlands which had been developing supramolecular polymers as biocompatible polymers, and had made out of these new materials cardiovascular implants. This company, Xeltis, in the Netherlands, used the supramolecular polymers for the surgical treatment of children which had a severe congenital cardiac malformation and required reconstruction of their heart. I can show you now the first young child who received this. This is here, Dominica. You see her here. Here she is 4 years old in 2013, and this was 3 months after the implantation on October 23, 2013. And it was done in the Bakulev Scientific Center for Cardiovascular Surgery in Moscow by Professor Leo Bokiria, you see here. You see, she looks very nice and she's smiling, and he's smiling too. Both feel well, huh? It's quite obvious. She has been saved, and he has done a good job. So they are both very happy. And of course, we were extremely happy also to see that this supramolecular polymer concept has led to materials which can be used for the reconstruction of the heart of children. Many more than 10 have now been operated on, and the same company has now also developed heart valves like the ones you show here, which have been also since 2016 have also been developed. This is a breakthrough in surgical practice, as I am told. Another property of supramolecular polymers is that materials made out of supramolecular polymers have an interesting property, which is self-healing. Here you can see a nice film. You see between the fingers of my co-worker here, you have this very nice film. In fact, incidentally, this was an Indian co-worker who is now at a company in Europe, it's BASF company, Dr. Nabarun Roy. So when he was in the lab, we developed this film here. Then somebody cleaves it with scissors. They cut. You superimpose the two ends of the two halves. You press on it with your finger if you want, and then you press for a minute or two, and you can stretch, and it sticks. So this is a self-healing property, because the interactions on which supramolecular chemistry is built, these interactions are labile, and if you cleave a material and superimpose it, they heal the cleavage. They stick together. They are a film again, which means it's a self-healing process. Okay, now let's go back to the main line that's pointed out at the beginning, that matter becomes complex by self-organization. I don't want to talk about the two possibilities, equilibrium and non-equilibrium. That's too complicated for this talk. So self-organization means you want to generate complex architectures by molecular information control building up. And this is a sort of a program which is present in these systems and leads to self-organization by design. Let me show you an example, which is an obvious one. It's just the build-up of a virus. A virus, and here I chose the first one which had been understood by Aaron Klug. And here you see on the left-hand side that this virus is made up of pieces, in fact, 2,130 bricks, which go together. And due to the shape and the interactions which link these bricks together, they form finally a sort of a tower with helical shape, a helical tower. And inside the tower is the genome of the virus, RNA. On the right-hand side, you see it in more detailed fashion. So this means here that the proteins, which are the bricks building up the virus, these proteins are built in such a way that they spontaneously interact with one another, bind together, and in an orientation which yields automatically. This is just automatic. This is not magic, nothing else. It's just automatic on the basis of the structure of the bricks, the interactions between the bricks, and a lot of some other properties. So we understand very well what is happening, but it looks like magic because spontaneously this builds up, but it builds up because it is designed for it. And this can be called a programmed chemical system. Then, of course, people were interested in trying to make artificial analogs of this type of process where matter self-organizes. And one way, I illustrated only in one way, taking bricks as molecules which can bind metal ions, like iron or zinc or cobalt and so on. So the ligand molecules are the bricks, the metal ions are the cement which hold these bricks together. And on this basis, we call them now metallo-supramolecular architectures because they're built on metal ions by using molecular bricks. And many were then made and designed in laboratories. We made double helix, triple helix. These are artificial ones, which have nothing to do with the natural DNA. But as you can see, that's a nice double helix. That's a triple helix. And this is automatic. You make the strand, you mix in the metal ion, automatically you get that. You can also make this type of grids where molecules are vertical or horizontal, and at each different points they are intersecting and held together at these points by metal ions, in this case silver, this here, Ag plus, silver. Circular architectures can be made, and even cylinders like this one, where you have 3 linear molecules in red, 4 flat molecules in blue, and 12 metal ions, which therefore are copper ions. When you mix together the molecules, the linear ones, the flat ones, and the metal ions, automatically you generate this nanocylinder. This is just to illustrate. Then one can play around, one can play with these properties, with the bricks, they have to be well designed, and with the cementing unit, which then brings them together. And if you do that right, you can generate on design this type of complicated, complex metallo-supramolecular architectures. So this is also of interest for nanoscience and nanotechnology, because ultimately you would like to generate spontaneously by design these supramolecular architectures in a well-designed way as an alternative or a complement to nanofabrication and nanomanipulation. Because it's quite obvious to everybody, I suppose, that if you can go from fabrication to have to make it, to self-fabrication, designing the bricks and the cementing units in such a way that they spontaneously will build up, this is the ultimate fabrication. And this is not done yet, but I think one can easily imagine that it will be possible to develop devices in this way. Now, let me close up with, not yet close up, but go to what we are now doing. I will show that just in a few slides. So, chemistry can be either molecular or supramolecular. And then you can design the objects so that by information storage, by programmation, they will generate in a controlled way functional, molecular, and supramolecular entities. So this is what I illustrated yet now, up to now. But then there is another step beyond this simple first step, which is beyond this design, there is selection. I mean by that if you have a complex mixture, then one can try to build it up in such a way that it will generate architectures, complicated architectures, by selecting the right bricks. And in order to do that, one needs a diversity of bricks, different types of bricks, and one needs dynamics. In other words, the bricks have to get together, but be able to re-separate, because maybe the first collision between the bricks is not the correct one. Correct meaning not the most stable in terms of thermodynamics, thermodynamical properties, so that they may attach to one another, but if it's not the best one of those which can be formed, which they available with the available bricks, then they have to go separate again and find something else. And this is then this is what we call constitutional dynamic chemistry, meaning by that the constitution of the chemical object is dynamic. It can break apart and reform. In this process, if the conditions medium change, then maybe then the resulting new object may be different from the one started with, which broke up first, and so it may adapt to changes in the environment. In other words, this leads to an adaptive chemistry, a chemistry where the entities which finally result are able to adapt to the conditions which are imposed by the environment. And this leads together to combine diversity, which is in fact recombining things, combinatorics, and of course, molecular recognition, which is information science, information, as I said earlier. Let me just illustrate that in a simple way. This constitutional dynamic chemistry allows adaptation by exchanging of pieces of molecules, and this has been used in a search for biologically active substances, for dynamic nanostructures, and also for developing dynamic materials, self-healing materials, responsive materials, and so on, which are, in terms of application, very interesting. And it develops then adaptive chemistry, which is our main theme of research right now. Let me just show you an example of how to use these processes, these type of entities, for a new process, a new procedure for developing drugs. If you have a series of components, which are shown here, and you mix them together, and there are different structures, different shapes, and so on. These are molecules, just represented schematically by these different types of colors and shapes. If they have here a black dot, which is a chemical reaction which can undergo reversible connections. So if you mix those together, what you get is what we may call a combinatorial library. That means all the possible interactions and pairing up and all the possible interactions between these basic bricks can occur. And so they generate then what we call constituents from the components, and these are always interconverting. So it's a very complicated mixture. So what can you do with it? You can use basic thermodynamics by when you add now a cavity, a molecule with a cavity, like an enzyme, then this cavity will interact better with will interact preferentially with some of these constituents, so that it will bind it and displace the equilibrium. This is for the people, for those of you who have done some physical chemistry. This is the principle of the law of mass action, the principle of Le Chatelier, which shows that if you introduce into an equilibrating mixture a given molecule which has this property of binding some entities in this mixture, it will shift the equilibrium towards the formation of that, which means we can express it in this way. The lock assembles its keys. And this is then a way to, a new way of doing drug discovery research. It's an adaptation to the receptor. One thing you might also be interested in, because that's an interesting material, an interesting properties which is given to materials, and I'd like to show you what can be done in the optical properties. Suppose you make two plastics, two polymers, two films, shown here in white and in yellow. And they are made from two components, A and B, or A prime, which is another way of A, and B prime, which is another of type B. Now, if the connections between the A and B, A prime and B prime, are reversible, then if you superimpose these two initial films by, for instance, here a corner, if these bonds can break, then A can combine with B prime, and A prime can also combine with B, which means that then you have at the places where you superimpose the possibility to generate the two combinations which were not present at the beginning. So if one of these new ones has a color or a fluorescence, you will see it. And let me just illustrate that. This was done with some Japanese co-workers. You have this cat, the head and the ears is AB. The inside of the ears, the eyes and the mustache is A prime, B prime. So if you now let it go, heat a little to accelerate, you can see that there is color and fluorescence at the places where the superposition occurs. In other words, in this process, there has been an exchange, a breaking of the AB and A prime B prime bonds, a recombination to generate the others, and one of these combinations had a color, yellow color, and the yellow-green fluorescence. This shows you in a way which is very convincing that indeed these materials have the property of changing because they are dynamic. They can rearrange their components. So chemistry, to summarize here, and then I want also to go into some more philosophical type of considerations. Chemistry started, of course, with atoms at the beginning. These are the bricks, and when you make molecules, then interactions between molecular systems, populations of molecules have supramolecular properties. Supramolecular chemistry is the chemistry of the populations of molecules. They can organize depending on the way they interact. Dynamic when they are weakly bound, so that they can exchange their components. They can adapt to given conditions. And these are the steps towards complex matter. Now, let me just insist now on some features which we have been seeing all the way in my talk, which are behind this, which are sort of on which this is built. You have noticed that chemistry can make new objects which did not exist before. So the essence of chemistry is not only to describe what already exists, but also to create new expressions of matter. In other words, the book of chemistry has to be not only read, but also has to be written, like the score of chemistry has to be composed before you can play it. So chemistry has a creative power, which makes it a sort of an art, like art. Everybody speaks about art, like creation. Chemistry is very creative also, as much as art, and maybe I would say even more intimately, because chemistry has this creative power on the insides of the object. It's the art of matter, the intrinsic properties. We heard about at the introduction that was also on the campus of MGM University. There was also an artistic section, Leonardo, dedicated, if I understand well, to Leonardo da Vinci. Now, Leonardo da Vinci was a fantastic person. He was somebody who is, of course, best known by the general public as an artist, by the famous Gioconda, Joconde. So this was one thing. But he was also an engineer and a scientist. And he wrote a sentence which is very, very profound in Italian. This is the Italian, and if you translate that into English, where nature finishes to produce its own species. We are part of that. These are what already exists around us in nature, as we call it. Man begins using natural things. Which natural things? These are, of course, the bricks of visible matter. This is the periodic table. These are the natural objects from which one can build complex entities in harmony with its very nature. This means, for me, at least, that it is in harmony with the laws of physics. You cannot go against the laws of physics. They determine everything. And then the ending is a very strong ending, considering that Leonardo da Vinci was a scientist, an engineer, and an artist. And for an artist, creation has a very strong meaning, which for science, I think, is the same. For me, I am convinced we should also consider that science is creating entities, creating new things. So to create an infinity of species, that's a very strong statement that mankind deals not only with what is natural, which already exists, but what is man-made. We have then to realize that the quest of knowledge, which is the goal of science, will lead us to the control of our destiny. It is knowledge which will give us the possibility to control our destiny. It is starting already. We have modified ourselves already. You may have an artificial lens in your eyes. You may have some titanium in your leg. You may have teeth, which are implants. This is not natural. This has been, many people are already modified. And so the path provided by science leads us from looking, increasing knowledge and understanding the world around us, and eventually to control ourselves, our destiny. I would like to cite a famous mathematician, David Hilbert, who once said in one of his talks and wanted that inscribed on his tombstone, two sentences. The first one is, wir müssen wissen, we must know. This is what drives scientists, we must know. This is what drives us to do the work, to try to have more and more knowledge. And then the second part is much more powerful, because it says that wir werden wissen, we will know. Not only we must know, but it's the conviction that through science we will know. I would like to finish my last slide. As has been said also at the introduction by Professor Jatinta, that science shapes the future of humanity. And I encourage all of you to participate, because your future, the future of your children, the children of your children, and all your descendants will depend on the development of science. Finally, the last property of science I would like to illustrate, because in the present modern world, with all these dangers, all these wars going on, science is an activity, and the only one mankind has. It's the only activity where on our planet, this is our planet Earth, this is a piece of dust, a very, very, very small piece of matter in an enormous universe. We are sitting on this. We have developed on this piece of dust, this ball turning around, we have developed science. And science is the only human activity which has no borders. Anything you know on the planet Earth is valid everywhere. A water molecule is a water molecule for us, it's a water molecule for anywhere in the universe, billions of miles away. Science has no borders. Science is not dogma. Science has no flags. Science has no borders. Science has no religion, no dogmas. Science is the same for everybody. And it's the only human activity which is like that, because what we discover scientifically is valid everywhere. So I would like to encourage all the students here to really consider that very seriously. Science brings everybody together and should, in principle, bring also mankind together. That is not yet successful. But if there's one activity which can lead to it, it is scientific activities, knowledge, science, and applications of science. So on this day of a Science Day in India, I am very happy to have been able to speak to you. You can ask questions if you want, and I wish you a very successful future. Thank you very much.
Thank you very much, Professor Lehn. A beautiful talk, a lot of knowledge filled in chemistry, in science, in so many things. I myself learned two words today, adaptive chemistry and self-fabrication. Frankly, I didn't know these two words earlier. So science is progressing every day. And we welcome first the students. If they have a query, don't be afraid. He will give a very simple answer to your questions.
Yeah, what I will do, I will now stop the sharing so that I can see the people, because when I talk with somebody, I'd like to see the people also, the students.
Sure.
So I will stop the sharing.
Please do.
And now I see you.
Yeah.
Voilà. That's good.
Can you see some of us there?
Yeah, I see the room.
Yeah, sure. Right. So now we see both the parts, right. Quite all. So this is the audience today's.
Yeah. Hello. Nice to see you. I would prefer to be there with you, of course, but all right, some other time.
Yeah. So who wants to ask the first question? He or she can come to this mic so that we can all see. And if necessary, I'm going to repeat that same question in our own terminology. Yeah, please tell your name.
Good morning, sir.
Good morning. Good afternoon for you.
Yes, my name is Prakash. I am the physics PhD student. First of all, thank you very much. Is a wonderful talk and very informative session for us all the students. So congratulations, and we are proud of you as a part of this Science Day. So I have the small question. So as you mentioned that the chemistry is nowadays a world so involved in the day-to-day in our practice and our the day-to-day life also. So can you give as a newcomer, as a student, what is the tips and tricks? So would you recommend the new researcher students and navigate their academic journey for effectively?
Will you repeat the last part of the question?
So new researcher student to navigate their academic journey effectively. Any tips and tricks?
He wants your suggestions.
Yeah, I understand. To navigate the academic journey. Yes, I understand.
Exactly.
Yeah, it's of course a good question. Pardon? Sorry?
Yes, and one more additional, you suggest that is mission and vision. So as when you get the first Nobel Prize in your research study, so what is the first reaction? Can you share your experience, sir?
Yeah, okay. This is an evident one. Of course, I was very happy. Okay, all right.
Thank you, sir.
That's the easy one.
Yes.
Okay, so how can you navigate an academic career? There is no simple answer to that. We are all different, so we can navigate a different way. We are in a society, different societies. We have education, different types of education, and we are all different. But there are some things one has to pay attention to, and especially if you want to do research. I think there are few attitudes which are important, which do not define exactly what to do, but the attitude with which you should operate. Let's say the attitude is first, don't miss the train. Now, what do I mean by that? Do not miss the train. That means if you do scientific experiments, sometimes the results, or at least if you work in science, sometimes what you are doing wants to tell you something which you were not looking for. So you should not miss that. Quite often it appears, especially in research, that there are events, there are occurrences which are something you didn't look for, but which just sort of happen. Now, in many cases, people discard them. They think, okay, this is not what I'm looking for, so I'm not interested. But sometimes those things are much more important than what you were looking for. So don't miss the train. Be attentive to everything that is happening. And don't consider that something has just to be thrown away. Maybe that is more important than what you were looking for. Then the second advice is, don't try to jump on a train which is already full. I mean by that, don't go for areas of science where there are already too many people. There are some areas in science where there are so many workers already that you will have not much chance to contribute a lot. Now, of course, the big advantage for those who do research, if you work in an area where there are many, many others working, you get many citations, you get recognized by many people, but you don't necessarily contribute something original. If one can, one should try to look for new parts, new ideas, which are different from what is already practiced. And the last advice, which I can, of course, there are many others, but I could give, is that try to think perpendicular in this way. Don't follow necessarily what you have been taught. What you have been taught in classrooms and so on, of course, is usually correct. Of course, it should be. But try to sometimes to reverse what you are accustomed to ask another question. And if you think against what you are accustomed to, you may be able to see something which is totally different from what you would like to see, what you know. But it is sometimes then, of course, shocking. It can be shocking because you look for things which are in opposition with what you have been taught, but it can bring you a lot of new ideas. Of course, it has still to be reasonable or rational. You cannot just do anything. You have still to limit this attitude to what is rational, scientifically valid. But it is a good way of trying to find some new areas worth developing and which would bring a lot of originality and creativity. So good luck.
Yes. So, who else has a question? Would you like to come to the mic and ask a question if you have? Come on. Please, tell your name.
Very good morning, sir.
Name?
My name is Dr. Suryakant Sapkar. I have one question. As a supramolecular chemistry having various applications in medicinal chemistry, can we use a metallo-supramolecular architecture as a conducting polymer? And another is, can we use these macromolecules in a membrane technology? Because nowadays we are leading in the e-vehicle technology. In a battery technology, can we use the supramolecules as a membrane?
Yeah.
Yeah.
Okay, so I mean the two questions are linked. Supramolecular entities are, in fact, molecules have always existed. Supramolecular entities have always existed. So the ones which use metals to bind things together, of course, are a special category. In fact, let me just to make things also sort of clear for those who are not chemists and so on, the difference between molecular chemistry and supramolecular chemistry can be summarized very, very simply. It doesn't mean that, of course, it's each field, molecular and supramolecular, is a complicated field, but it can be summarized very simply in the following way. If you look at a water molecule, a single molecule of water, all of you know what it is, H2O, H-O-H, okay? A single molecule of water cannot boil and cannot freeze, has no viscosity. So a glass of water is also water, but a glass of water can freeze, it can boil, it has a viscosity, it has an index of refraction, and so on. What's the difference? The difference is that you have a lot of water molecules which all interact in a supramolecular way by forming what is called hydrogen bonds. So the difference is shown there as from isolated molecules. There's the world of isolated molecules where they are defined by their structure, their shape, and so on. And when you have populations of molecules, mixtures, then you have, of course, new properties which emerge. So this happens then progressively. Complexity, my title of my talk was Steps Towards Complex Matter. This is a sort of the first step is going, let's say the first step is really from atoms to molecules. This is molecular chemistry. The second step is populations of mixtures. And this is supramolecular where things interact. And of course, then this can become more and more complicated. I think it's too long to go into details. And in fact, we are still far away from understanding life, the origin of life and the origin of thinking, which is much more important than life even. For me, the thinking is much more important than living. If you live, okay, that's all right. But if you don't think, you are very limited. So I would say that when these supramolecular entities, and especially the ones which have midlines and molecules, they can be, of course, used for different kinds of applications. Not so much has been done yet. Some have been done. I showed you these supramolecular polymers, which were used for different applications, which can self-heal or can be used in biomedical applications. But there are others, and I showed you some applications in the course of my lecture. Do you have you want another compliment to that?
Hello,
No, sir. Thank you very much.
Okay,
Thank you.
No, he's happy.
Okay, thank you very much. That's good.
Yeah. You
have a nice lecture room.
He's going to ask you a question.
Yes. I am Professor Vilas Sapkal, Vice Chancellor. Background is chemical engineering.
Aha.
I work in University of Ottawa and Göttingen University as well. In the journey, as you rightly explained, the mechanism of complexity, atom, molecules, supramolecules, but the stage you mention is formation. And they get organized, they form, and then the complexity starts. Is there any mechanism defining the way of formation? How do you control it, understand it, or they do it on their own? Or what is the explaining mechanism of formation or coming together this supramolecule and creating the complexity?
Yeah, you are an engineer, you do chemical engineering. I think what molecules do is exactly what you do on the macroscopic scale. You have your building blocks, which are atoms, and you can, out of these atoms, make molecules which have a given shape. And then you have to understand what happens when they come together, when they interact. And for instance, I showed you some of the things we have made, these metallo-supramolecular entities, where metal ions bring together organic molecules. This depends on the way the organic molecule is built and which metal ion you use. Metal ions, for instance, like iron, they have 6 points of attachment. They form an octahedron. And if you have now an organic molecule which can bind to that, then it will be arranged by the rules of the way in which iron 2, for instance, interacts with the molecules. So this is determined. It's like, this game which children use, at least in Europe, called Lego. You know the Lego game? Okay. Yeah, the Lego game has, let's say, geometric rules. You can do something with that, but not everything. If the pieces have a given shape or given points of interaction or given ways to put them together, you will not be able to generate things which do not correspond to these ways of putting them together. And that is the case with atoms. I took the example of an iron atom, an iron ion, like iron 2. This iron 2 wants to form, by interaction with molecules, by forming an octahedron around itself. And you cannot make something else. It forms an octahedron, point. So this is the thing where you have, like when you make a building, if you make a building, you use bricks. And if you have a building of a given shape, you have to use bricks of a given shape to build them together and generate a building. So I think making complex molecules is a way of engineering where you use the atoms as the cementing units, or let's say where you build molecules which can serve as bricks for different for generation of different architectures. But pardon, it's just the interaction is just an attraction which comes from electrostatic attractions or repulsions, and this then determines what comes out. There are rules in that which you cannot escape.
Yes, surely. But that has to do with thinking, design, and building. So,
yes.
As you rightly mentioned, that it is still difficult to explain the mechanism of thinking.
Oh yeah, this is still very far away. Sure. Thinking is still, this is the ultimate frontier. Huh? The ultimate frontier is what is thinking. People have been thinking about thinking for a long time.
And what is the interface between like supramolecule or this process and the living cell? What is that interface from where this non-living is starting living?
Yeah, this is a good question. That's where we don't understand it yet, but we know that life is based on molecules. Life is based on the way molecules interact, so it's molecular and supramolecular. But of course, I try to explain by taking this example of an isolated water molecule and a glass of water, the fact that that it's not so simple maybe, but the fact that you have a population of water molecules as compared to an isolated one brings in new properties which did not exist on the level of a single molecule. And this sort of makes you think that as complexity of a mixture or complexity of systems increases, new properties will appear. And one of them is life. Now, how does it occur? This is still something which is far away, but okay, you know, we understand more and more all the complexities. If you are a biologist, you can sort of find fantastic these complications of living organisms, which nevertheless is able to live. It's so complicated. But the problem is that when we ask this question about life, we usually think about humans. Humans are, of course, extremely complicated. They are at the top. But they are very simple molecules, very, very simple entities. Bacteria, they're also living. It's much, much, much more simpler. So we have to understand progressively the steps. We cannot jump from molecules to a human being. That's too far away. So we have to go from molecules to population molecules, then new properties appear, and eventually trying to understand how, for instance, a bacterium functions, which is much simpler, but already very complicated, of course. I think we should not jump over. We have still a lot to do.
Thank you so much for your explanation. Thank you.
Shall I ask your comments on two things? One is, James Allison, he played with the checkpoint protein so that the immune system does not think that the cancerous cells, which have gone rogue, but they are the usual cells are like the immune system. So that is one thing which is called possibly the chemical engineering of a molecule to do the right kind of drug delivery or therapy. Number two, your compatriot, the French physics Nobel Laureate, Pierre de Jeamp, he tried to tell everybody, I don't like my work to be called complex matter. I would like it to be called soft matter. I think he was simply
what matter?
Complex matter to
soft matter. Yeah, sure.
So
yeah.
colloids, polymers,
sure.
and surfactant. I think he was simplifying the knowledge of the building blocks, which are these colloids, surfactants, and polymers, for the benefit of young students. So they should, particularly physics students, they should not be afraid. So that is the thing. So can you comment on the first one, for example, James Allison?
Jeamp was one of my colleagues at the Collège de France, so I knew him pretty well. We have a lot of discussions together. And indeed, I mean, I think the difference is, I think it's the same, because soft matter is a way to describing the fact that it is not a solid, not a hard matter. Huh? So that's a sort of a mechanical property. Complex matter means that you have and this is something which for a physicist is a bit, the physicist should know a bit more biology, because in biology it's very, really complex, meaning lots of functions, lots of things where everything is interlinked, where one process talks to another one, where there's a feedback. So it's, when you look at some of the biological processes, even simple things like a Krebs cycle and this kind of things, this is very complicated, and one wonders how it can work, but it works. Since it works, it must be understandable. Huh? I think if it works, we as human beings, we work. Huh? The thing works, not always, but I think with time it's degraded also. But it works. And this is something one has to try to understand, but it's very complicated. So we are part of soft matter in some ways, not the bones. The bones are hard matter. But we are soft matter, but very complicated way with many processes occurring, controlling one another. What is amazing is the fact that one of the most basic fundamental processes is, of course, the genome. And the genome is very simple: 4 letters and 2 ways of interacting. That is really sort of at first it's startling. But on the other hand, maybe in the evolution, more complex systems have been tried, but it was chaotic. It was too many errors and so on. And so what survived in evolution was the one which was the simplest. Good enough, huh?
Yeah. Thank you. But you mentioned DNA computing, for example, because DNA can store information, it can process information.
Yes, exactly.
So, I mean, just a bet, long shot. Do you think DNA computers will win over quantum computers?
Sorry, that's DNA what?
You have quantum, I'm a physicist. So
Yeah.
I bet on quantum computers. Quantum computers. Quantum computing. But
yeah, this is an interesting point because, this is something very fundamental you're asking, which is that quantum computers do not exist naturally. And there are other things like that which mankind can make, which do not exist naturally. And so this is this power of science to use the principles we discover in what exists to go further than what exists already. And this is also what Leonardo da Vinci said. Of course, he didn't know about quantum computers, that's for sure. But in fact, quantum physics is a very, very strange thing because there are properties there which are not part of our macroscopic world, the way we live, but which are there. They are there. There's part of it.
You said that dark matter, dark energy, which comes on the quantum side,
Yeah, I don't know for the moment. I'm not a cosmologist. They have to tell us what it is.
Thank you so much. Really, it has been a very enriching experience for so many students who are still sticking out here through our complicated discussion as well. So let us all give him a big hand because he.
Thank you very much.
We were very fortunate to listen to you in a really good talk and even the question answer session. Thank you so much. You want to say?
What is fantastic with this way of sort of speaking together? Of course, I would have preferred being in Aurangabad.
Oh, yes, I
For
want
sure.
Yes.
But on the other hand, we can talk to each other, we see each other, and so it's not too bad. It could be much worse if we had not the video makes it very nice. And in fact, I see you, I see the students, so that is already a fantastic step bringing us together.
Right.
But we look forward to your visit to MGM University at Aurangabad.
You should come, he says.
Yes, we extend a very warm it's invitation.
Thank you very much.
Next
time, if I come to India, I will let you know.
Yes, thank you so much for accepting our invitation. Thank you.
Anybody else?
Thank you very much for the amazing lecture. It was very interesting and knowledgeable. A very beautiful explanation about molecular and supermolecular chemistry from beginning to the present scenario. Your lecture will definitely benefit our research scholars, sir. Thank you, sir.
Thank you very much for the invitation and for having the possibility to talk with you and with the students. And I wish all of them very successful future.
Thank you.
Thank you, sir.
Thank you.
We close this session. Thank you.
Thank you.
Bye-bye.
Bye.
Thank you. Bye-bye.
Now I invite Dr. Vilas Sapkal, sir, Vice Chancellor, MGM University.
Good luck.
Thank you. Close.
I invite Dr. Vilas Sapkal, sir, Vice Chancellor, MGM University.
The recording has stopped.